Hello, everybody, and thank you for having me at the British Atherosclerosis Society. And thank you to Hella and the co-organizers for inviting me. I will share my screen now. And share the slides. And go straight into my presentation, which will be a review from vignettes that we've mined so far from our work on cell atlases of human organs with respect to insights into the vasculature. And as I'm sure you're all aware, here in the Sanger Institute, about 5 years ago or so, we rallied the international community to come together and start working on atlasing of human cells. And this is really a collaborative effort to create a comprehensive reference map of cells and tissues. And of course, this is driven by fundamental curiosity as well as applications to diagnosis and disease. And the catalysts really that made clear that this would be possible were the technical advances in genomics, the resolution revolution in genomics that allows us to measure the nucleic acid content of single cells, and in tandem, ways of profiling tissue sections using sequencing as well as imaging. And so together, these technologies integrated with computational methods are on a trajectory to create a Google Maps for the human body, where we go from a sort of coarse-grained view of cells to high resolution, full molecular breadth information in terms of a sort of street maps view of cells in tissues. And this Google Maps of the human body will be built by the global community, and there are now over 2,000 members from 76 countries from around the world, many hundreds in the UK. And if you're interested in joining for a particular biological interest or technological interest or computational methods, everyone is welcome to join at humancellatlas.org/join-HCA. And it's a very easy signing up process. That means that you get the newsletter, access to technology discounts, provided that you sign up to a few basic principles, including open data sharing, ethical principles, principles of inclusion and diversity, and so on. So where we stand at this point in time is that we've got data from many of the major human organs and tissues to a quite extensive level. And overall, there's at least some data from 22 different tissues where we count about roughly 50 at an initial level overall. And this is a figure from a review by Rosa Elmentaite, Cecilia Dominguez-Conde, Ling Yang, and myself that's in preparation. In revision, and Rosa Elmentaite is the PhD student who assembled this figure. And it gives you an impression of the information about tissues. And of course, every single one of those contains cellular components of the vasculature, both the cardiovascular and the lymphatic system. And so, so this data, in a way, provides a rich playground for mining information about these systems. And of course, there's huge medical importance to understanding the vasculature, as we're obviously in the meeting of the British Atherosclerosis Society. And this includes atherosclerosis, but also many other diseases specific to individual organs. And in addition, there's of course the relevance for understanding and predicting drug delivery and how this could change upon, for instance, disease information and so on, in terms of localization and accessibility of drugs. What I want to do in the next 20 to 30 minutes is ask what we've learned from the perspective of my own group by mining this data with respect to cell states. What are the cell states in the vasculature? That's not just the endothelial cells, but also the surrounding smooth muscle and parasites. And how heterogeneous are those cells? Can we say anything about their developmental and regional relationships, and what are their signaling networks? And I want to say a special thanks to Michaela Noseda, who's a long-term collaborator on the heart, but also vasculature studies using cell atlas technologies, and also Lu Yang in my group, who's been responsible for this integrative sort of survey of cells that I'm going to present here, which is very preliminary, but I want to share with you sort of where our thinking stands. So in terms of using this data to establish how heterogeneous vascular cells are, there are some known cells and known markers that emerge, unsurprisingly, but also new ones that have come out. And this is from the whole community and the cell atlas literature over the past 5 years or so. And so that arterial endothelial cells are marked by FNB2, and then you've got the smooth muscle cells around them, capillaries with Claudin-5, RGCC, and venous cells, where the marker ACKR1 has emerged both from the murine and human literature using single cell technologies that wasn't known before. And in terms of parasites, there are and endothelial cells. We obviously have continuous fenestrated and sinusoidal vessels and endothelial cells accordingly with different cell states, and also parasites, different cell states of parasites that I'll come back to. And so using the unbiased genomic data integration approach, mostly, that's at the moment, mostly stands at the level of single cell RNA sequencing data and single nuclear RNA sequencing data, we can establish cell types that are shared across tissues and found as common states across many different tissues, and they include the ones that I've just mentioned, but also organ-specific vascular cells. So for instance, in the heart, endocardial cells, which I'll come on to, marked by NP3R SMOC1, and identify new markers for these. And then alveolar and pulmonary endothelial cells that have emerged from data mining of airway data from the community. And so these, I'll present these in a bit more detail later. If we look at the heart, which focusing on our data on 6 regions of the heart that was published in Nature last year, there are many different endothelial cell populations that we're able to identify from the data, and they're shown in the different colors in this UMAP representation over here on the right. And they include arterial, venous, capillary cell states, as you'd expect, and as well as smooth muscle and parasites over here on the left-hand side. And by integrating these data sets using our cell phone database in terms of cell surface receptors and ligands, we're able to predict different modes of interaction of the arterial endothelial cells with the arterial smooth muscle cells, with certain sets of notch and delta ligands interacting with each other, whereas the venous endothelial and smooth muscle cells are predicted to interact with a different set of receptor ligands binding the cells together. Now, what Ling has done is integrate data from many different tissues, including the heart and lung that I've just talked about, but also many other organs and tissues, skin, kidney, basically covering, as I said, about 22 different tissues. We're covering 11 organs, sorry, with many different tissues and hundreds of samples. And what this is obviously has various technical challenges from a computational point of view, given the complexity and size of the data, and they include removal of ambient RNA or soup, standard quality control, removal of doublets, batch correction, and correcting for technical noise, but maintaining biological noise. This is obviously a huge challenge and homing in on the vascular cells, leaving about 200,000 cells. What this revealed was a couple of tissue-specific populations. So I've talked about the populations that are shared across tissues, but there are certain populations that are specific, and that includes the heart endocardium, as we'd expect. And if and that's a population that's very clearly specific to the heart and separate from other populations. And Ling worked with James Cranley and Kazumasa Kanemaru in my group to tease this information out and map it onto spatial transcriptomics data that we have using 10x Genomics Visium technology for the free wall of the chambers of the heart, right atrium, left atrium, and apex. And this gives us confidence in our annotation that we have endocardial cells localized, predicted from suspension cell data and localized to the correct place in the Visium data. You can see this sort of computationally colored in blue here, for instance, in the apex, in this region, in the right atrium, in this region of this tissue section data, that's about 0.5 centimeter squared. And then other cells, epicardial fat, myocardiocytes, and so on, in the other colors. And this, we can check basically that we have a statistically significant enrichment of these particular endothelial cell population in the endocardium using the cell to location framework from Omar Bayraktar's group, developed with Oliver Stegle by the PhD student and colleague Clef Chevnikov. And that allows us to very elegantly integrate the suspension cell and nuclear heart data that we had that was published last year with this new spatial transcriptomics data that's unpublished, and basically establish this cell population in the endocardium in a statistically rigorous and unbiased way. If we look at the skin, we find rare skin-specific endothelial cell populations. And one that I'd like to highlight is one that we published earlier this year with Mas Hanifa's group and Fiona Watt's group, that are SNCG positive endothelial cells. You can see it's a minor population within the endothelial cells in this pink group over here. And they're marked by expression of cytokines, chemokines, signaling to leukocytes much more than the other endothelial cell populations. And so they sort of caught our attention from that point of view. And when we compare their abundance in healthy skin, we see significant expansion of this population in the autoinflammatory disease samples that we studied in the same work, atopic dermatitis and psoriasis. And when we predict which cells that endothelial population is interacting with, unsurprisingly, based on the cytokine chemokine expression, it's immune cells, in particular this macrophage-specific, skin-specific macrophage population. And so we predict that venous endothelial cell population that's signaling to the macrophages plays a role in the response of the skin in autoinflammatory disease. And so in terms of the cell states in the vasculature and how heterogeneous they are, I hope I've given you an initial impression of common cell populations in terms of arteries, capillaries, veins that are found across all tissues, but then also specific, tissue-specific and organ-specific endothelial cell populations that we've identified. And the ones that I mentioned were heart endocardial cells and the skin immune signaling macrophage interacting endothelial cells. We can also interrogate developmental and regional relationships. And I just want to give one small vignette from ongoing work with Michaela Nazeda's group. So there are obviously gradual changes in gene expression along the anatomical axis when you're comparing arteries to arterioles, capillaries, venules, and veins. And we can quantify those relationships basically when we take, for instance, heart endothelial cells. So this is a tissue that we've been working on together over many years. And I just want to indicate here work from Michaela's group, Alek Husinov, calculating and quantifying those relationships between the endothelial arterial cells here on the top left, the capillary cells, and then the venous cells. And whether that's a purely zonation or developmental relationship, I think is an interesting and still open question. But what the utility of this is showing in a quantitative way changes in particular molecules over time. IGF binding protein, family member is high in arteries and then decreases as you go to the capillaries, and then is essentially off in veins. There are other markers that come off in capillaries, and there are others that are specific to veins. I mentioned ACKR1 as a new marker that's come out of this single cell genomics literature, but there are also others, like as shown down here. And this is a sort of unbiased way of establishing the compartments, and also the gradual changes across these compartments from the arteries through the capillaries to the veins. In the final section, I want to go into a bit of detail of how endothelial cells interact with surrounding cells, or also vascular cells interact with other compartments, including myocytes, the immune system, and so on. So, and that includes interesting and important roles that the vasculature plays in immune trafficking. Going back to the organ that's been a focus here, which is the heart, in that spatial transcriptomics data, in those tissue sections that I mentioned earlier, Kazumasa, James, and collaborators are able to manually annotate from a histology point of view large vessels and the perivascular regions. And of course, the transcriptomic data here is charted in 50 micron spots. So we have 50 micron diameter features from which we can read out the transcriptome in these particular histological regions. And what this allowed Kazumasa and James to do is to basically define co-localization of particular compartments like cardiomyocyte populations with specific vascular cells, and to compute this co-localization in an unbiased statistical framework. And what this shows is co-localization of a particular cardiomyocyte population that's in the ventricles. We call it VCN3, and that's localized to specific regions. It's not spread homogeneously throughout the free wall of the ventricles, but it's clustered in specific regions. And we find statistically significant co-localization with particular arterial smooth muscle cells, adipocytes, and fibroblasts. And when we look at the interactions between those specific endothelial cell subpopulations of the arteries, cardiomyocytes, and fibroblasts, what it suggests is that there's a fibrotic cell signaling circuitry that could be going on. And so here we're for instance, charting the signaling with VEGF-A between the endothelial cells and the ventricular cardiomyocyte, that specific ventricular cardiomyocyte subpopulation out of the 5 different ventricular cardiomyocyte populations, and the interaction between fibroblasts and the smooth muscle cells, for instance. So there's basically bidirectional signaling going on that's happening through the FGF signaling axis and VEGF signaling, suggesting a sort of circuitry between angiogenesis and fibrosis that may be going on here. I also want to share with you unpublished data on the lung, from Elo Madison, who's a postdoc in the group, supervised by Kirsten Meyer. And what they've proceeded to unravel here is the cell populations in 5 distinct regions of the lung, going from the trachea to large airways and down to parenchyma, small airways and alveoli. And again, this allows us to dissect the different endothelial cells, smooth muscle, parasite, and so on populations, and then pinpoint the specific localizations of them. And what this reveals is particular venous endothelial cells this time. So in the heart, it was arterial endothelial cells that I just presented in terms of the cell signaling circuitry. Here, they're venous endothelial cells. And these particular venous pulmonary and systemic endothelial cells have a very distinct leukocyte homing signature, where they have interaction partners on different immune cells, ranging from B to T and NK, and cells in the myeloid compartment. And these interactions are shown, the receptor ligand interactions are shown here, again using cell phone DB, and the endothelial immune interaction here. A similar pattern is evident for airway parasites, and this is the parasite cell population shown down here that's secreting chemokines that are sort of calling and recruiting, again, these different immune cell populations. So you've got CXCL12, CCL2, CXCL21, CCL19, sort of these canonical chemokines that are recruiting immune cells and possibly contributing to extravasation. So in summary, I've given you an impression here, I hope, for what we can learn in terms of vascular cells and their interactions from human cell atlas data from specific organs and tissues. And in the first part of the talk, I was asking, how can we use this data to find out what the cell states are in the vasculature and how heterogeneous they are? And I presented Ling's unbiased approach to integrating many, many data sets and then identifying organ-specific populations. And I presented some data from that, from the heart endocardium, and then also the published skin immune signaling population. I presented markers that we can mine in terms of their expression across artery, capillary, venous zonation and developmental relationships. And then finally, I presented cell signaling networks from unpublished heart and lung analyses, where we find indications and predictions of angiogenesis and fibrotic circuitry in the heart by integrating single-cell RNA sequencing data with spatial transcriptomics data in a computational framework using cell-2 location, and then also finding evidence for leukocyte homing endothelial cells in the airways. And I want to finish there and thank you, and I look forward to the Q&A. And I want to emphasize again that the amazing teamwork in all of this. And I've mentioned Michaela Noceda in Piro, who's a long-standing collaborator on the heart and the vasculature, and Ali Huseinov and Sam Barnett from her group, Ling Yang from my group, and also the unpublished work and analysis on the heart from Kazumasa Kanemaru, James Cranley, and Elo Madison, Kirsten Meyer, and many others in my group. I'd also like to thank other collaborators for analyses and sharing data along this journey: Mirjana Efremova, Rosa Venter-Tormer, who are both former postdocs in my group, who now have their own labs, Mas Hanifa, Mena Clark, and Cora Said Parsi in Cambridge, and support teams from within the Sanger Institute. And thank you. I'll take any questions.